We will move on to chapter 27, which is about bacteria and archaea. These are two other branches of the three main domains, the third being the eukaryotic cells or eukaryota. Um, and these two major branches of the domain, triumvirate, the three parts, are vast. They're huge groups of different life organisms, and so what we will do is we'll talk about them generally. We will talk about some of their functions, and then we'll go into a little bit of specific, but not too much. We're just talking about the difference between bacteria and archaea, in terms of their cell wall, in terms of the way they replicate, some of their behaviors in terms of whether they colonize, etc., and then go into a little bit of specifics. But both of them are highly adaptable, and it's one of the things that has made them so successful on the planet Earth is their adaptability. Some of them can live in pHs that are acidic enough to dissolve metal, which is pretty kind of amazing to think about. And some of them can live in environments so cold or so hot that no other organisms have been found living. And it's just amazing their adaptability to a wide range of not only chemical differences but heat differences, and even some with regard to oxygen or lack of oxygen, and some of them are adaptable to live in nitrogen atmospheres. They adapt very well to normal habitats too. Don't get me wrong, they're not only found in extremes, but normal habitats such as lands, waters, and their range of adaptability to different habitats is why they are so abundant. Also, they're very small. They're about 1,000 times smaller than a eukaryotic cell, and so they are microscopic. There's only one that's about as big as a poppy seed, and that's an archaea that's about as big as a poppy seed. In terms of individually, now not in terms of a colony. They first were thought to inhabit the Earth about 3.5 billion years ago. Their long history in terms of evolution has is what leads to their diversity today and their adaptability that we find today. Most of them are unicellular, so one-cell organisms, but some of them tend to stick together. We're not even talking about colonies because some of them can colonize, but some of them tend to stick together almost like we think of as tissue, as we think of cells that get to stick together to make tissue, like our tissue we have in our body. And remember, much of this, going back to what we learned in chapter 1 with this concept of emergent properties, and certainly when we talk about things, that then may have emerged into what we identify as part of our physical being, it makes it very interesting. Now, I'm not saying that our ability to create tissues came from this particular property, but you can definitely see where there may have been a pathway from eukaryotic cells, instead of being unicellular, to have just a couple of them stick together. Now, we also colonize into much bigger units. Here in the image you have your three major domains, and then you also have a typical bacteria cell. A couple of things that we'll talk about in the course of this chapter. We'll talk about the fact that the genetic material, DNA, is not encapsulated. It's not held in a membrane. You have various things sticking off the the outer membrane of the bacterium. One of the longer ones you see here is called a flagellum. That allows this bacteria to have some motility, to have some movement. You also have smaller things sticking off, the small red ones called pilus. Some of those pili allow this bacteria to stick together, allow it to have adhesion to other bacteria or adhesion to other cells. One particular pilus that we'll talk about is called a sex pili, and that allows for DNA or genetic material to pass from one bacteria to another. You also have a cell wall, so a plasma membrane. That's the white inner membrane, plasma membrane, and we've talked about that a little bit in previous chapters. Plasma membrane allows certain things to get inside the cell, other things to not get inside the cell. And then inside the plasma membrane you have cytoplasm. So cytoplasm, DNA, plasma membrane, all of that sounds pretty familiar, as well as ribosomes, actually protein areas where proteins are produced. One of the things that's a little bit different is this capsule. We have a little bit of familiarity with cell wall, that wall being a structure that is more supportive, I guess, or withstands more different forces than that plasma membrane. But this capsule can serve many purposes with regard to a bacteria. It can make the bacteria much more resistant to temperature changes, to changes in pH, by just protecting the inner DNA of the bacterium until that particular environment shifts, or until this bacteria somehow or another, basically making an endospore. We'll elaborate on these things as we go forward. There are basically three different types or shapes. The most common shapes, these aren't the only shapes, with regard to prokaryotic cells. They're divided, and so just to repeat, prokaryotic, and I guess we're just introducing this word right now, prokaryotic. Remember, we talked about the difference between eukaryotic, prokaryotic. Prokaryotic tend to not have membrane-bound organelles. They tend to be smaller. They tend to have a less complex genetic makeup, which we call a genome, so a less complex genome than eukaryotic cells. And two major domains, archaea and bacterium, or bacteria. A difference between bacteria and bacterium is whether you're speaking plural or single. They have great diversity in terms of their genes, and in terms of those genes being expressed in what they look like. They thrive almost anywhere, as I said. Acidic environments, and we're talking about serious acidic environments, cold, hot, where you would maybe not find any other organisms. Most are microscopic, most are unicellular. Some can colonize, so some can stick together, and generally colonization happens for mutual benefit. It's better for the bacteria to have more than one in an area than just one. Much smaller than eukaryotic cells, and the three main shapes are farthest to the left is spherical, and what you think is so a circle. When you see those, think of streptococci, or think of diplococci, and you probably have, if you haven't had a sore throat, one of your children have, and the doctor probably says, well, that my child has strep. What you have is a spherical shaped bacterium that has colonized in your throat, streptococci, or staphylococci. So strep or staph many times cause throat problems. The middle one is a rod-shaped, and with regard to rod-shaped, we call it bacilla, or rod-shaped. And those are usually solid individuals, but can form some chains. And not as common, but Streptobacilla is a disorder. And the third one to the right is a spiral shaped, and these spiral shapes can be more like a comma or a corkscrew. And spiral shapes also, unfortunately, can be problematic in some disorders. So 3 main shapes. And then cell surfaces, and I apologize, this is cutting off at the left. I will try to be more of a reader for you with regard to that. But the cell surface, and we're talking about the capsule. We're not talking about the plasma membrane, we are talking how the capsule layer attaches to the plasma membrane. And what this allows the actually, I think I'm going to stop this right now and fix this slide so it's not cutting off. We'll pick this up on the next one.